Hello and welcome to IB Biology Notes with Dr. Kimo. This is A4.1 Evolution and Speciation. Standard level, mostly about evolution, and higher level, mostly about speciation. Evolution is the change in the heritable characteristics or traits of a population. Many traits are heritable, and over time, some traits tend to become more common in a population. Their frequency increases. Meanwhile, other traits become less common. Their frequency decreases. What you see on the right is an imaginary population where blue and yellow are common colors and large is a common size. Time passes and environmental conditions change. What we get in our example is more red individuals and more small individuals. The frequency of red has increased, the frequency of small has increased. Also, mutations have introduced a new color and a new shape. Changes in the frequency of traits can be due to natural selection or random genetic drift. More about this in another chapter. Some traits may disappear altogether, and new traits may appear through random mutation. Now, natural selection, random genetic drift, and random mutation, they work together and they have no aim, meaning evolution has no aim. Charles Darwin published his work on the origin of species in 1859. He described how natural selection is the mechanism behind evolution. Alfred Russel Wallace independently developed the same basic idea, but Darwin became more famous over time. Before Darwin and Wallace, there was a famous naturalist called Jean-Baptiste Lamarck, who was an early proponent of biological evolution, but he suggested a different mechanism, namely inheritance of acquired traits. In a famous example, the original short-necked ancestor of giraffes is stretching its neck to be able to reach leaves higher up on trees. When this giraffe has calves, the calves are born with a longer neck. The calves keep stretching their neck, and when they have calves, those calves are born with an even longer neck because of all this stretching. And this goes on generation after generation, and the neck of the giraffe becomes progressively longer. However, this mechanism is not correct. Let's look at some pieces of evidence for evolution. First, comparative genomics and proteomics. Already the fact that all living things share the same genetic code suggests that all life is related and has evolved. The change in heritable characteristics is due to a change in DNA sequences, and the more similar two species are, the more similar are their genomes. Modern genome-based cladograms or family trees correlate well with older morphology-based classification. And gene families are shared across species. A gene family is a set of several similar genes. Taken together, the similarities and differences are such that the only coherent explanation is the splitting of lineages, i.e., evolution. Another piece of evidence is selective breeding. Humans have selected animals and plants with desirable traits for thousands of years. And today, all domesticated animals and crop plants are the result of a long history of selective breeding. Examples include livestock with high milk and meat production, non-aggressive dogs with herding or hunting instincts, and fast-growing crops with large edible parts. On the right, you can see a wild mustard plant, which has given rise to cabbage, kale, cauliflower, broccoli, and Brussels sprout. One more piece of evidence, homologous structures, which are similar due to shared ancestry. A good example is the pentadactyl limb of vertebrates. Comparing the limbs of vertebrates points to a common origin. Now, looking at these examples below, You can see that the limbs have changed over time as species have adapted to their respective environments. This development is called divergent evolution. And homologous structures are found throughout the animal and plant kingdoms, but they are not always easy to spot. Analogous structures, on the other hand, have evolved independently. For example, the eyes of vertebrates to the left and of cephalopods to the right have developed due to similar selection pressures, often in similar environments. You could be fooled to think that they share a common origin, but their anatomies reveal that they don't. A classical example of a difference is that in the vertebrate eye, the retina is behind the nerve fibers, and in the cephalopod eye, the retina is on top of the nerve fibers. In vertebrates, to the left, this leads to a blind spot, which does not exist in cephalopods. Analogous structures are the result of convergent evolution. Let's move on to speciation. When a population is split by a geographical barrier, the two groups tend to develop in different directions due to different conditions and random change. This illustration shows a population of non-flying insects which is split in two by a geographical barrier, namely a river. The conditions on each side of the river are not identical, and over time the two populations develop in different directions. After enough time in isolation, they develop into two distinct species. This process is called speciation, and it requires reproductive isolation, meaning that the two populations are not able to reproduce with each other. Now we are moving into higher level territory. Geographical separation is probably the main reason behind reproductive isolation and speciation. And speciation due to geographical separation is called allopatric speciation, different homelands. Speciation can also occur when a population splits in two populations that continue to inhabit the same geographical area. Then we talk about sympatric speciation, same homeland. Sympatric speciation may be caused and upheld by temporal separation, where animals mate or plants bloom in the same area at different times, or behavioral separation, where animals prefer different niches or have sophisticated courtship behavior, for example. Here's an example of sympatric speciation. Two groups of the same species of cichlid fish live in Lake Masoko, Tanzania. One feeds in deep water, the other one close to the shore. They have adapted to their respective environments and prefer to mate with fish from the same group. This has led to behavioral separation, and if it continues, it will lead to sympatric speciation. A population may split and spread to occupy different niches. As the groups adapt to their new environment, speciation may occur. Sometimes this happens rapidly and repeatedly, and we call it adaptive radiation. Charles Darwin described the adaptive radiation of birds on the Galapagos Islands in his work on the origin of species. Let's consider some barriers to hybridization or mixing of species. Hybrids often have a lower fitness than either parent species and an enhanced risk for genetic anomalies. Hybrids are often sterile, especially in animals, and if fertile, hybrid individuals may reduce the frequency of species-specific adaptations. Producing hybrid offspring can therefore be a bad or risky investment, and so there are barriers of hybridization. For example, complex courtship behavior, that pollen doesn't germinate, or that gametes don't fuse. Now take a minute to study the graph on the right. It shows how Arabidopsis seeds, which are hybrids, are often collapsed and rarely germinate. The last topic of this video is polyploidy. Most eukaryotes have two sets of chromosomes. They are diploid. But some plants have 3 or 4 sets of chromosomes. They are polyploid. Polyploidy is usually the result of whole genome duplication, which is duplication without cytokinesis. In crop plants, polyploidy often has advantages. Polyploidy can lead to rapid speciation. Let's start with autopolyploidy, more than 2 chromosome sets from the same species. Imagine that a diploid plant becomes tetraploid. D means 2 and tetra means 4. This tetraploid plant will produce diploid gametes. If they fuse with the haploid gametes of the original plant, the result is an autotriploid plant, which is infertile. So the diploid plant and the tetraploid plant are essentially separate species. Allopolyploidy, more than 2 chromosome sets from different species. On the right, we have 2 species of diploid plants. They both produce haploid gametes. These 2 species are rather similar, so the haploid gametes are able to fuse, but they produce a hybrid plant which is infertile. However, if this hybrid plant undergoes polyploidy, what we get is an allotetraploid plant, which is a new species. It cannot reproduce with either of the original plants. That's all for now. See you next time.